Greetings, dear learners. We were learning analytical toxicology. Up till now, we have finished different extraction techniques before the final detection of drugs and poisons, that is, pre-treatment of the biological matrices, before the final analysis of the analyte. So, in the last class, we learned the classical and modern methods of the extraction. For the gases, we perform microdiffusion, adsorption, desorption. Then modern method that includes the gas analyzer, gas chromatography, volatile inorganics, Gutierrez method, Mass Spectroscopy method, microdiffusion, dilution with specific reagents under the specific condition of the pH, crystal test, color test. Then the modern technique for the volatile inorganic is the microwave oven technique for dilution, followed by ion chromatography using ion exchange resins, spectroscopy. For volatile organic compound, distillation, steam distillation, diffusion, then gas chromatography, mass spectrophotometry. Then for non-volatile inorganic compound, dry and wet incineration, group analysis, electrodialysis, dilution under the appropriate analytical conditions, tetrametric, paper and thin layer chromatography. Then modern technique for the non-volatile inorganic includes the microwave oven technique for dilution, followed by ion chromatography using ion exchange resins, inductively coupled plasma atomic emission spectroscopy, that inductively coupled plasma mass spectrophotometry. Then under the non-volatile organic solvent extraction, STAS auto method, dilution with ammonium sulfate, sodium tungstate and the related method. Modern method for the non-volatile organic include HPLC, paired ion extraction, chromatography, HPTLC, supercritical fluid chromatography, solid phase extraction, Michelin extraction, affinity chromatography, microwave assisted reaction system, accelerated solvent extractor, sweep co-distillation, universal trace residual extractor. In for the anions, dialysis, chemical digestion, paper and thin layer chromatography. In the modern technique, ion chromatography by ion exchange resins. We discussed most of the techniques in brief details. Some of them we will be discussing along with the analytical process like the HPLC, TLC, like the mass spectrophotometry, these things, ion chromatography, these things in the detail we will be talking in the analytical methods. Now at least you have a brief idea that what steps are followed in the toxicology lab for the analysis of the toxins. So if I summarize whatever we have learned till today, so there is a specimen that has been collected from the body and that is subjected to the preliminary test like the color test, bedside test. Once the preliminary test diagnoses some suggestive of the something, so according to that the further analytical processes are done. So if the compound is non-volatile, then we categorize it is non-volatile or this is volatile. If it is volatile compound, so it is collected through the head space method, so that it will be coming in the gaseous head space chamber, where it will be collected with using gas chromatography, and then this will be further this up till here it is the extraction and all those method that has been applied. After the gas chromatography and HPLC, then for the detection analyzing the analyte or solute, the mass spectrophotometry will be used. So this will be the step for the volatile compounds. Then for the non-volatile compound, again categorized into inorganic and organic. For organic, we have the extraction methods, immunoassays. Then extraction method includes the liquid-liquid extraction, solid phase extraction, SPME and various other techniques that we talked already. Then that will be subjected to the fluorescent infrared spectrophotometry, then TLC, then HPLC. Then for the inorganic compounds, X-ray fluorescence analysis, that in then the infrared chromatography, then atomic absorption spectrometry, then the ICP-AES, these all are the followed for the inorganic compounds. So in this way, this whole steps of the toxicological analysis in the analytical toxicological lab are done. Then coming to the analytical processes. So, for the analysis of poison in the forensic science laboratories or the labs, the various analytical methods are applied that can be categorized into the qualitative and the quantitative method. Qualitative methods include the chromatography, whereas the quantitative methods along with the qualitative methods can be through the spectrophotometry. Under the spectrophotometry, different types of spectrophotometry: absorption spectrophotometry, emission spectrophotometry, mass spectrophotometry. They will be studied in the detail. Then chromatography, then immunoassays, then chemical analysis, certain chemical reagents that are applied and that is used for the analysis of the poison. Then neutron activated analysis. We will study the analytical methods in these steps. That important thing here to highlight is that combination of the gas chromatography and the mass spectrophotometry, that is GCMS, is considered the gold standard of the toxicological testing because it is 99% specific and 99% sensitive. Best analytical technique that has been till date available is the GCMS, that is the combination of the gas chromatography and the mass spectrophotometry. Then coming to the spectrometric methods. Terms spectroscopy, spectrometry and spectrography are often used interchangeably. But there is a fine difference. A spectroscopy is the direct viewing of the spectrum, that is, scope means the viewing. A spectrometry is the measurement of the spectrum, that is, metron, that means measure by an analyzer. Then a spectrography is the photographing a spectrum, like in the mass spectrography, from the spectrum and the photography. In the practically, there are two types of spectrometry. One is the absorption spectrometry and the emission spectrometry. In the absorption spectrophotometry, absorbed rays are analyzed, and in the emission spectrophotometry, emitted rays are analyzed. Mass spectrophotometry is not the true spectrometry in the sense that neither absorbed nor emitted light is analyzed. In instead, a spectrum of molecular fragment is analyzed. So, mass spectrophotometry is basically a misnomer. In general, emission spectroscopy is more sensitive than the absorption spectrophotometry. You can see here in the picture, this spectrophotometry is based on when light is passed through any compound, it emits certain spectrum of light that when analyzed, it may be help to identification of that substance. So, this is the idea behind the spectrometric methods. So, it can be done in two ways, that once a certain light or emitter is passed through an object, it may absorb certain of the components of the spectrum. So, by analyzing that what spectrum has been absorbed by the compound, we can say that this belongs to a particular group of compound or this is a particular compound. And then in a similar way, in the emission spectrophotometry, at the atomic level, when the charged ions from the higher level goes down to the lower level, then there is emission of the spectrum. Once we detect that spectrum, we can say this belongs to a particular molecule or object. So, this is basic idea behind the spectrometric methods. Then coming to the absorption spectrophotometric methods. When the light is passed through a substance, it absorbs certain specific frequencies depending upon its molecular structure. By analyzing the missing wavelengths from the emerging beam of light, an inference about the nature of the substance can be made. This is the basic principle behind the absorption spectroscopy or the spectrophotometry. It can be of various types. One is the infrared spectrophotometry. Infrared spectrometry is used when the substance absorbs wavelength in the infrared region. So, the steps for the infrared spectrophotometry is that first the sample shall be properly prepared, that is by pre-treatment, extraction and the cleaning up procedure. Then for the gaseous samples, they require a sample cell with a long path length to compensate for the diluteness. Liquid samples are sandwiched between the two plates of a salt like NaCl, potassium bromide or calcium fluoride. Solid samples can be prepared in a number of ways like KBR pelleting process. Sample is grounded with the KBR finely to remove the scattering effect from the large crystals. Once the sample is prepared, then a beam of infrared light is passed through the prepared sample. From the emitted light, an absorbance spectrum is produced. It shows the infrared wavelengths which the sample absorbed. Analysis of these absorption characteristics reveals details about the molecular structure of the sample. As you can see in the picture, this is the infrared source that is passed through a chamber that contains the sample, and this sample then scatters the certain lights or that absorbs certain light that is presented over the detector, and then it detects that this particular spectrum is missing, so it got absorbed. So the label will be telling the what amount of the absorption and what emitted rays has been absorbed, and depending upon that we can also make the quantitative assessment of the compound in the solution. Then UV spectrophotometry. UV spectrophotometry, because UV light source is used, also known as ultraviolet visible spectroscopy or the ultraviolet visible spectrophotometry, because visible light is used. Principle is similar to that of infrared spectrophotometry, except that here UV rays are used in the wavelength of the 200 to 400 nanometers. The procedure for the UV spectrophotometry is that a light source emits the light. Once the light gets emitted, dispersion of the light into the individual wavelength is done by the prism or the diffraction grafting. This selection, then the selection of the single wavelength is done by the appropriate filters. The entrance slit dispersion device or the filter exit slit are together referred to as monochromator. So from the condenser to the prism to the slit is called as the monochromator. So this from the light source, this condenses the light, or that is the convex lens that may be used on the prism. This prism then splits into the different spectrum, and this slit lamp that circulates the UV spectrum, and that is bombarded over the sample of interest, and from there the UV spectrum absorption is noted. That particular spectrum is absorbed. From this we can identify the compound of the interest. So the sample is placed in a transparent quartz cuvette in the path of the UV rays which has been selected. The amount of UV rays which transmit through the sample is measured by the detector photocell. Monochromator can be so set up that it sequentially allows UV rays of the all wavelengths. Transmission of the each wavelength is recorded by the photocell. A spectrum is prepared from the resultant data which can be studied to derive the chemical composition of the sample. The UV spectrophotometry is mainly used for quantifying the blood levels and for the drugs like paracetamol, salicylates, and the urine level of phenothiazines compound. Then atomic absorption spectrophotometry. This works on the principle that when a ray of light is passed through a sample, electron of the atoms can absorb energy and be promoted to the higher orbitals. That is atomic absorption. The absorption will cause one specific wavelength of light to be missed from the transmitting light because it is absorbed by the atom. All the elements absorb a specific wavelength of light only, depending upon their unique atomic structure. Just as a person can be identified by his fingerprints, an element can be identified by the specific wavelength of light it absorbs. Elements in the sample is identified by analyzing the missing wavelengths. This is the basic concept behind the absorption spectrophotometry. So here this atomic absorption spectrophotometry, the atom absorption is occurring at the atoms level. So you can see in the picture here the sample is taken and reflected with the cathode lamp, and that spectrum that is seen over the using analyzer and then the computer screen. Then this will show the absence of any spectrum that is suggestive of that compound. So, as the number of atoms in the light path, the amount of light is absorbed is increased, that in that proportion. So, by measuring the amount of light that has been absorbed, even a quantitative determination of the substance can be made. Such kind of determination are more commonly used in toxicology. For this stage, the sample shall be reduced to the atomic level for the proper analysis. The atomization of the sample is done by breaking the chemical bonds and releasing the individual atoms. This is done by increasing the temperature of the sample. This can also be achieved by two other methods like flame atomic absorption spectrophotometry and without a flame spectrophotometry, that is the using the electrical furnace, that is called FAAS. The advantage of FAAS over the AAS are that it has higher sensitivity and its features of the temperature programming, and that is control of the temperature. And its unique feature is that it has the temperature programming, that is under it temperature can be controlled. Then remaining spectrophotometric absorption spectrophotometric method can be microwave or Mössbauer spectroscopy. In the microwave region of the electromagnetic spectrum is used, whereas in Mössbauer spectroscopy, gamma rays are used. Mössbauer spectrophotometry was invented in the 1950s by the Rudolf Mössbauer. On that name, this became called as Mössbauer spectroscopy. In both of these, emission spectroscopy is also possible. So, absorption as well as emission is possible with the microwave and the Mössbauer spectroscopy. Then coming to the emission spectrophotometric methods. As already talked, that here the emitted spectrum is analyzed, and as the emitted spectrum when passes through the any object or the compound, it has the lesser strength, so that decrease in the spectrum strength or the other features will help to analyze the emission spectrophotometric methods. Under that, fluorescence spectroscopy is one of the method. Here, it is a type of emission spectroscopy that analyzes fluorescence from the sample. A beam of light, usually UV light, is passed through the sample that causes molecules to emit light of lower energy than the typical visible light. And the analysis of emitted light gives the nature of the sample. Devices that measure the fluorescence are called fluorometer or fluorimeter. So, here you can see this laser is giving the light and the sample is reflecting the emission, and from there we will be analyzing what type of fluorescence is there, and then according to that we may decide what compound it belongs to. Then another emission spectrophotometry is the inductively coupled plasma atomic emission spectroscopy. So, atomic emission spectroscopy is the reverse of the atomic absorption spectroscopy. In this technique, wavelengths of the light are studied which are emitted by the atoms or the molecules during their transition from an excited state to a lower energy state. That is, in the atomic absorption spectroscopy, an incident light falls on the sample, its molecules absorb some wavelength. Analysis of the emitted light by the detector reveals their nature. However, in the atomic emission spectroscopy, molecules within the sample falls to a lower state. They emit the light which is analyzed to reveal their nature. So, the principle of atomic emission spectroscopy fundamentally consists of three general steps, that is the breaking of the sample into the constituent atoms, atomization, excitation of the atoms, and the emission as electrons fall back into their original orbits. So, of these, the first step, that is atomization, is used in atomic absorption spectroscopy as well. Atomization should be produced with a minimum ionization of elements to be analyzed. It can be done by a number of methods, like by heating the sample to a higher degree. Inductively coupled plasma atomic emission spectroscopy utilizes plasma as the atomization and excitation sources. Plasma is a state of matter similar to the gas in which particles are ionized. So, atomization should be produced with a minimum ionization of the elements to be analyzed. It can be done by a number of methods, like by heating the sample to a higher degree. So, on that basis, one of the method of the emission spectrophotometry is the inductively coupled plasma atomic emission spectroscopy. Here in this emission spectrophotometric method, the atomization is done by the plasma. So, ICP-AES utilizes plasma as the atomization and excitation sources. Plasma is a state of matter similar to the gas in which the particles are ionized. Plasma can be produced by supplying energy to the matter. The sun, lightning, and aurora borealis are the examples of the plasmas found in the nature. An inductively coupled plasma produced by the supplying energy in the form of electric currents produced by the electromagnetic induction. For the creation of the inductively coupled plasma, argon gas is typically used to create plasma by the electromagnetic induction. Its temperature ranges from the 4000 to 8000 degrees Celsius. Use of this plasma as an atomization and the excitation source, sample is introduced inside the plasma. Its molecule immediately collide with the electrons and charged ions in the plasma. Samples is broken down into its respective atoms, that is atomization. Electrons in the individual atoms get energy from the plasma and rise to a higher level. As they return to their original position, they emit radiations. In the process, they give off the radiation at the characteristic wavelength of the elements involved. This radiation is studied to derive the composition of the metal. It can detect numerous metals simultaneously based on their emission lines. Non-metals like phosphorus, sulfur and halogens also have a strong emission line in the vacuum ultraviolet light. Then the Raman Spectroscopy. This is also a type of the emission spectrophotometric methods. Here in the 1923, American physicist Arthur Compton showed that when X-rays are scattered by the matter, the wavelength of the reflected X-rays are increased because of the electrons absorb some of energies of the X-ray. So, based on this principle, in 1928, C.V. Raman showed that the same phenomenon occurs with the visible light also. This is called as the Raman effect. Raman spectroscopy involves the passing the visible light through the drug sample or analyzing the wavelength of the transmitted light. This discloses the structure of the drug molecule. This has been used for the knowing the composition or profiling of the several drugs like the MDMA, ecstasy. So here you can see in the picture, this is a picture of the machine in the lab, and then the method, the light is passed through the sample and then it has some emission, and that is reduced emission analyzed in a spectrophotometry, and that is presented and the molecule is identified. This is the basic concept behind the Raman spectroscopy. Then another spectrophotometry technique is the nuclear magnetic resonance spectrophotometry. It is also a type of emission spectrophotometric methods. Here it makes the fact that nuclei of the some molecule absorb radiation at the characteristic frequencies in a strong magnetic fields and then radiate them at the characteristic frequencies. By identifying these characteristic frequencies, identification of even teeny or impure samples is possible. So you can see here in the picture, the sample tube is radiated with the magnet and then it detects the radiation that has emitted by the sample, and that will be analyzed using the spectrophotometer. Then after absorption spectrophotometry and the emission spectrophotometry is the mass spectrophotometry. Mass spectrophotometry is an analytical technique that detects poisons first, breaking them into the molecular fragments and then measuring the mass to charge ratio of each molecular fragment. Although it is a misnomer that as the light spectra are not studied in it, so a spectrophotometry term is wrongly used with the mass spectrophotometry. However, it is the most reliable identification methods for the poisons and the drugs. However, its applicability is only if the sample is pure. In case of impure samples like the admixtures, The sample is first subjected to the fractional separation by the gas chromatography. The procedure is then more commonly known as GCMS, gas chromatography, and mass spectrophotometry, that we already discussed is the best method for the analyzing the poisons. So the steps involved in the typical MS procedure are loading of the sample. The suspect sample is loaded onto the MS instrument and it is vaporized. Then it is ionized. Then components of the ionized sample are impacted with the high energy electron beams. It results in formation of the charged particles, that is ions. Other methods for the ionization of the samples can be chemical ionization. Accordingly, the name could be the chemical ionization mass spectrometry, done by the chemical means with the ions of a reagent gas, usually methane, ammonia, and isobutane. Then another technique could be the electrospray ionization mass spectrometry, that is ESIMS. Then after ionization, then the third step involves the separation of the ions, that is done by the magnetic field, that is ions are made to move in the electromagnetic fields. So you can see here in the picture that sample is once loaded inside, then it is vaporized, and then ionization is done, and that is moved through the magnetic field, and then according to the movement, according to their mass and charge ratio, finally the detector will detect the ions, and ions are detected on the analyzers. On the basis of this analysis, they are processed into the mass spectrum. So this mass spectrum is an intensity with the mass to charge ratio. Plot various lines represent the molecular fragments and their height and their respective concentration. By studying the various lines and their height, the poison present in the sample can be inferred. So you can see here in the picture, this is the mass spectrum picture. So a molecule that has the this mass to charge ratio, that is heating at the 500. So this has highest this quantity. So by comparing, this is a typical pattern, or you can say DNA fingerprint like a pattern. From there we can analyze that this product is this compound or not, because the different compound will have different types of spectrum produced. By studying this, we can analyze the compound that has been present in the biological matrices. Then, as we already talked, that the ionization process can vary. Ionization can be done by the chemical methods, by the electrospray ionization. Accordingly, the name of the mass spectrophotometry will be the chemical ionization photometry, electrospray ionization photometry. So this electrospray ionization mass spectrometry is recently developed technique, especially useful in analysis of the macromolecules, because it overcomes their propensity to fragment when ionized for its development. John Bennett Fenn was awarded Nobel Prize in Chemistry in 2002 for it. It uses electrical energy to form the ions. Here, neutral compounds can also be converted into the ionic form. Steps for the ESIMS is the first dispersal of the fine spray of charged droplets, followed by the solvent evaporation, and then ion injection from the highly charged droplets. Within an electrospray ionization, a continuous stream of the sample solution is passed through a stainless steel. This you can see, this ESI capillary, where it is passed through a continuously, then which is maintained at a high voltage relative to the wall of the surrounding chambers. A mist of the highly charged droplets, charged parent droplets with the same polarity as the capillary voltage is generated. A nebulizing gas, especially the nitrogen, sears around the eluted sample solution that ensures the increased sample flow rate. The charged droplets generated at the exit of the electrospray tip pass down a pressure gradient and potential gradient toward the analyzer region of the mass spectrometry. Another stream of nitrogen drying gas and increased ESI source temperature further decreases the size of the charged droplet. This leads to an increase in the surface charge density and decrease in the droplet radius. So the movement will be more faster. Finally, the electric field strength within the charged droplet reaches a critical point at which it is kinetically and energetically possible for the ions at the surface of the droplets to be ejected into the gaseous phase. So, by this way, and then further the mass spectrophotometry that we discussed, in a similar way it will be analyzed and it will be presented on the spectrophotograph. So, by this way we analyze samples using spectrophotometry.